Good morning students, I am Dr. Samrina, Department of Biotechnology, Central University of Kashmir. Today we are going to discuss transportation of proteins inside the cell. How proteins are transferred inside the cell. As we know, the eukaryotic cell contains two main components, cytoplasm and nucleus. This nucleus has again four components, nuclear annular, chromatin material, and nucleolus and nucleoplasm. This chromatin material contains the information which are coded into the proteins like amino acids. Now this chromatin has to be transcribed in the mRNA or RNA. Then this RNA moves out of this nucleus into the cytoplasm where it has to get translated on the proteins, either the free ribosomes of the cytoplasm or the ribosomes which are present on the endoplasmic reticulum. Those are called as rough endoplasmic reticulum. The nuclear annular, another is the nuclear annular, fourth component of the nucleus. Its membrane is interconnected with the endoplasmic reticulum, and endoplasmic reticulum's membrane is interconnected with the Golgi bodies. The whole interconnected membrane system is called as endomembrane systems. Now, in case the other organelles which are present in the cytoplasm are mitochondria, Golgi bodies, plasters in case of plant cells, lysosomes, and many other different organelles. In case of eukaryotes, there are mostly three types of transport systems: gated transport, transmembrane proteins, transmembrane transport, and vesicular transport. In case of gated transport, it is the transportation of material between the nucleus and the cytoplasm and vice versa. In case of transmembrane proteins, it is the transportation of material from cytoplasm to plasters, cytoplasm to mitochondria, or either cytoplasm to endoplasmic reticulum or cytoplasm to peroxisomes. And the third type of transport system is vesicular transport. It is between the endoplasmic reticulum and Golgi bodies, or from Golgi bodies to the endoplasmic reticulum, or from Golgi bodies to the late endosomes or to early endosomes, or Golgi bodies to the secretory vesicles. These are the three different types of transport systems. As we can see in this diagram, different types of transport systems: gated transport, transmembrane transport, and vesicular form of transport. Now, the DNA is transcribed into RNA in the nucleus. Then this RNA moves out from the nucleus into the cytoplasm where it gets translated. Now the translated protein, it has to see its destination. For its destination, they should have signal sequence attached either at C terminus or at N terminus. Like if we take example of peroxisomes, they are having three amino acids for peroxisomes. These are serine, lysine, and leucine at C terminus. When the sequence is present at the C terminus, then this protein is destined to the peroxisomes. And another is mostly these signal sequence are 3 to 80 amino acids. They are having different types of sugars attached to it or either phosphate group. For endoplasmic reticulum, these sequences vary from 5 to 15 amino acids, or in case of mitochondria, they vary from 20 to 80 amino acids. Now, when this protein is translated in the cytoplasm and they are having attachment of sorting signals, now sorting signal will decide to which organelle it has to move. Now, I have taken the example of protein import by mitochondria. This is one of the examples here. There are many different receptors which are present in the mitochondria, but I have taken one example: Tim complex and Tom complex and Tim complex. Now, in case of mitochondria, the precursor proteins are having attached with signal sequence. They are also attached with heat shock protein. These are also called as chaperones, heat shock protein 70. So it means the heat shock protein is attached here. They don't allow this protein to fold. This signal sequence is first recognized by the Tom complex, which is present at outer mitochondrial membrane. When this signal sequence gets inserted into the space, then the another complex comes into the action that is called as TIM23 complex, which allows the passage of cytoplasmic proteins from outer mitochondrial membrane in the inner mitochondrial membrane. Then TIM23, another complex which is the part of this TIM complex, comes into action, which allows this protein to form 3D complex in the mitochondrial matrix space. Then the signal is attached to this protein also there only. Then signal peptidase come into action. They cleave this signal sorting signals which is attached to this protein. Then signal peptide is cleaved from the mature protein. Then another protein come into action which is called a heat shock protein HSP60. HSP60, which allows the proper folding of this protein inside the mitochondria. This was the one example how proteins are imported by the mitochondria. Now the second is proteins transported to peroxisomes. As we know, peroxisomes are single membrane organelle. They don't have their genome, as in case mitochondria are having their own genomes, peroxisomes don't have their genomes. So all the material, all the proteins needed for the peroxisomes are coming from cytoplasm only. As the peroxisomes are having many oxidative enzymes, these are catalase or ureate oxidase. They play important role in our body, as we will see later. Peroxisomes are having many enzymes, as I said, catalase or oxidase ureate. Peroxisomes use molecular oxygen to oxidize organic substrate inside the cell. Mostly these peroxisomes are present at kidney or liver, where they detoxify harmful chemicals which are present in our blood. These peroxisomes use molecular oxygen which help to remove hydrogen from the organic substrate and gives hydrogen peroxide. This hydrogen peroxide is utilized by the enzyme catalase enzyme, which again removes hydrogen from other organic substances and release water and releases water. So this type of reaction is called as peroxidation reaction. This peroxidation reaction is important in the liver and kidney, as it removes harmful chemicals from the blood. Now when these proteins are formed in the cytoplasm, which are assigned to peroxisome, should have sequence at C-terminal. These are serine, lysine and leucine. When this sequence is present at the protein at the C-terminal, then this protein will be transferred to the peroxisomes. Before transferring this protein to the peroxisomes, they are recognized by the soluble receptors which are present in the cytoplasm. These receptors are called as peroxins. These soluble receptors are called as peroxins. 6 different types of peroxins have been identified till now. These are PEX5, which are important, PEX5, PEX1, PEX6 and PEX7. These are different peroxins. Now the protein which has to be transferred to the peroxisomes first will combine with the PEX5. When they will combine with the PEX5, they make a bond formation. Then this PEX5 plus protein which has to be transported to the peroxisomes make a bond. They come to the lumen of the peroxisomes. They come to the lumen of the peroxisomes where PEX1 and PEX7, PEX6 come into action. They break this bond which is formed between the PEX5 and the protein. by the utilization of the energy from the ATP. Now this PEX5 go ubiquitination, and then this PEX5 is sent back into the cytoplasm and protein get transferred into the peroxisomes. If it has been studied that if there is deformity in the peroxisome, they lead to severe disorders, abnormality in the brain, liver or kidney. Brain, liver or kidney, and these children die soon after birth if they are having deformity in the peroxisomes. Scientists have said that if there is mutation in the gene at PEX5, then severe type of abnormalities are created. And if mutations occur at PEX7 gene, then mild types of symptoms occur in the person. Now the third example is transportation of proteins in the nucleus. As I have made a diagram earlier where I have shown nuclear pore. These nuclear pore are having 3.5 nanometer radius. They are basket-like structure and are having cytoplasm, different 8 cytoplasm filaments which are towards the cytoplasm of the cell. And then another is the nuclear ring which is having core towards the nucleus. Now the protein which has to be transferred into or out of the cell should have specific sequence to pass through the nuclear pore. For the import of the proteins, these nuclear proteins should have nuclear localizing sequence. These are basic amino acids, essentially doublets amino acid, 5 to 14 upstreams. And the second component of this should have the important alpha and important beta. The important alpha is composed of P54 protein or P55 nuclear localizing sequence receptors and karyofurin alpha. Then the important beta is made of P57 and karyofurin beta. When these components are present for protein import, then the process takes place. Now this is the handmade diagram. This is the protein, karyophilic protein, which has to be transferred from cytoplasm inside the nucleus. It should get first attached with the important alpha. When it gets bonded with the important alpha, then another important comes into action, important beta. They form a complex, triple complex. When this complex is formed, they get interacted with the cytoplasmic filaments of the nuclear pore, which allow the passage of these proteins inside the nucleus. When the whole complex is transferred into the nucleus, then they get split up. As in case of nucleus, there are two different types of transportation systems. First is free diffusion, or second is active type of transport. Free diffusion is taking place when the molecule is having size less than 40 kilodaltons. If the macromolecule is having greater than 40 kilodaltons, then active type of transportations takes place. Now second is the mechanism and regulation of nuclear protein pore complex. Here proteins are moving out from nucleus into the cytoplasm. This is nuclear export sequence. It gets attached with the proteins. Then this protein makes a complex with the exportin 1. When this exportin 1 gets attached, they makes a complex. Then another RAM GDP comes into action. They form a triple complex. Then the whole complex is transferred out of the nuclear pore into the cytoplasm, where this RAM GDP gets converted into RAM GDP and exportin 1 and the protein along with the nuclear exportin sequence. This is how the proteins are exported from nucleus into the cytoplasm. This is the things which we discussed now was the proteins which are transcribed on the free ribosome of the cytoplasm. Now another example is when proteins are translated on the ribosomes which are present on the endoplasmic reticulum. This pink is our mRNA. it gets translated on the ribosomes which are present on the endoplasmic reticulum. First, when this process, translation process happens, endoplasmic signal sequence get recognized by the signal recognition particle. When this signal recognition particle get attached on the receptor which is present on the membrane of the endoplasmic reticulum, then they allow this chain, amino acid chain, into the lumen of the endoplasmic reticulum. Then the signal recognition particle and its receptor gets dissociated from the endoplasmic reticulum. Then the growing polypeptide chain is in the lumen of the endoplasmic reticulum. Then via the vesicular transport system, these proteins are transferred to the different organelles. Now, targeting of proteins to the lysosomes, as first the proteins are translated on the rough endoplasmic reticulum, then vesicles are coming to the Golgi bodies which gets fused with the Golgi apparatus. Then these proteins get addition of a phosphate group to the mannose group on the polypeptide chain. Then these polypeptide chain along with the mannose receptor form a bud-like structure which are having coat of Catherine. When this whole process, when this bud get detached, pinches off from the Golgi bodies, they form a vesicular type of membrane or system. Then they get attached with the lysosomes. Then they deliver their protein to the lysosomes, and again this receptor is again forming the bud-like structure which pinches off from the lysosome and get decycling, and again this membrane is getting attached with the Golgi bodies. This is how vesicular transportation takes place. This is another example. This is another example how this transportation takes place. This is our donor membrane. This is our lumen. Hydrophobic and hydrophilic ends are there. Hydrophobics are mostly towards the outer side. This is the proteins, transmembrane proteins. When these proteins, the proteins which are present in the lumen get oriented in a protein bud-like structure, then this bud get pinches off, make a transport vesicle. Then they start docking with the membrane of the donor's recipient cell. They get inserted into the membrane, and whole transport which was transported by the vesicular system get inserted into the recipient cell. because ज़्यादा तेज बोला मैंने